that we are talking about two things common thing, that is one is the drug, that is the ligand, another is the protein, that is the receptor. If we are knowing the ligand, then there's no problem. That means we are knowing the drug, and we have to know the receptor. If both things are known to us, this is a general fact. This is a general thing which the 99% students are doing or researchers are doing. They are having the receptor, known receptor. With that, they are just downloading it through the uh, protein database data bank, that is PDB. There are different, by the different IDs they are available. Different proteins are available on the basis of the disease or the problem which we have. Through that, it was just downloaded. From there we can have the active site, and the through that active site we can just, if the structure of ligand is known to us, we can generally just dock it over that particular receptor. That is known as drug receptor interaction, and this is an example of rational drug design and structure based drug design. So this is the most common way of drug designing which almost all pupils are using, that is known as molecular docking. Another, that is nothing but when we are having the known structure and their receptor or the protein is not known to us, that is known as QSAR or pharmacophore modeling. What we do here is, that was used in 1960. You can see that QSAR modeling were done, and over that, through that we design certain pharmacophore, and we are knowing that pharmacophore. By designing the pharmacophore, we dock it over certain receptor, and we try to calculate out its different activity, that this drug may be active over certain disease. So this is nothing but indirect drug designing and ligand based drug designing, and this may also known as analog designing, that is involve the 2D 3D QSAR and pharmacophore modeling. Another is that if we don't know any ligand as well as the receptor, if we are not known to anything, that we are just having that we have to develop any drug, and we are not knowing that what is the receptor involved, what is the ligand involved. So what we do, we are having high throughput screening combinatorial chemistry. That is nothing but, we can say, uh, it is a way by which we are using the blind screening of the drugs over the targets. And here we use the homology modeling. Homology modeling is used to design the receptor. Through that, over that receptor, the site were designed, the active site were designed, and over that the drug is going to dock over that. And final, we try to conclude any decision that whether the drug is active or not. If we are having any receptor which is known to us, but we are not knowing the ligand which are responsible for that particular ligand, so what we do, we are, that is known as receptor based drug designing. Receptor based drug designing, because we are knowing the receptor, we are knowing the active site, so that is why it is known as receptor based drug design. This is also known as de novo design. This is also nowadays it is used because we know that we are able to identify the disease, we are able to identify the protein, we are able to know that which enzyme is responsible for its activity, so we can easily take out that enzyme or that protein, and on that protein we can dock the different drugs. That is also that we have used in the case of COVID itself. That was the receptor based drug designing, de novo designing, to design out the different drugs. In that, the quinine has come out, or different drugs has come out which were like tetracycline was evolved out. Okay, that they are the drug, they are active over that. But not only the this designing of the drug, apart from that, these are the just a step by which we can design the drug, but we have to go through the different steps of the designing, in which the ADME as well as the dynamics of the drugs are studied. Then finally in vivo studies were done. There, from then only the drug molecules come out. So this is a basic, uh, all the basic modeling strategies which we are using to design any drug. This is one of the important slide for the drug designing. If we want to conclude the drug designing in a single slide, we can conclude with this slide that we have the different ways of modeling strategy, that are drug receptor interaction, receptor based drug designing, that is structure based drug designing, ligand based drug designing, homology modeling. So these all terms up can you can make clear from this slide. Now you can see that computer-aided drug designing technique involve the ligand-based drug designing, structure-based drug designing that maybe use the pharmacophore perception, QSAR, docking, scoring. Apart from that, we have to do the pharmacokinetic modeling. We have the Swiss ADME model. We have different software by which we can easily calculate out the uh, their pharmacokinetic parameter. That has to be also see. We have to also see that parameters also. Then the chem informatics, that is the database management. These all are the different techniques of computer aided designing. Now just we discuss the what is the QSAR and how it can be done. So you can see that QSAR is nothing but when we know we have the structures with us, we have the drugs with molecules with us, or probable molecule with us with their biological activities. That means the drug molecule with the biological activities are known to us, and with the help of the molecules which we are having and their biological activities, we decide certain parameters, that certain physiochemical parameters, and we design certain molecules on the basis of the existing molecule. Then we develop one pharmacophore, and that pharmacophore will have certain variables, and that variable will help us to design the new drugs. You can conclude it like this, that we are having the compound with the biological activity, then we go to the QSAR and we will develop the new compound with the help of the known compound which of biological activity. So we will develop the new compound, then we see its biological activity in the lab. That is the approach which is used by the QSAR. So you can see here that this diagram itself is showing that we are having the virtual structures. These are the proposed structure of any compound, and this is the library of the compound. Library proposal is nothing in the QSAR. They are the known compound. They are the compound which are already reported. Their biological activity is reported. Through that, we design the drugs which are having the same which may be having the same biological activity, but they are designed on the basis of the existing molecule. But we have the limitation with the QSAR that we require a big series of the drug, because small series of drug if you will take, it will not give the better result, it will not give the precise result. After that, you can see here that stage by stage the quality assessment to reduce the costly late stage iteration. This diagram is taken from the Nature review. From there, it is said that when we decide or when we take any lead, how it come out from multiple leads, it comes with the refinement, and finally we require the regulatory development. So it require this that has a large attrition rate. If we will not go through the lead identification, it will reduce the attrition, it will reduce the reduction, that is the rejection of the drugs if we go through the clinical, that is rational drug design. This QSAR can be divided into two types, two-dimensional or three-dimensional QSAR. Earlier there were only two-dimensional QSAR. Nowadays we have the three-dimensional QSAR along with the 2D QSAR. So you can see in the QSAR what is the assumptions, that is the we have one known compound which is having the known molecular activity. On the basis of the biological activity which has, we design the certain molecules, then we select certain parameters. That is, one parameter is the dependent parameter, another independent parameter. So the dependent parameter is nothing but the parameter which is already known to us. Suppose we are having a molecule like triazole series we have taken, and its antibacterial or biological activity in the case of antifungal is given. So we'll take the IC50 value, that is the dependent parameter. Then we decide that we have to take the physiochemical parameter, different parameter we have to take, so that will become independent parameter. So through that we will going to run the through the software, and we are going to conclude out it in the form of equation. So that equation will going to help to predict the pharmacophore. So we can say these are the steps of QSAR. First of all, we have to select the training set. Training set is taken from the research papers. Then we have to enter the biological activity. That activity is also taken from the that research paper. That is over that already the in vivo activity is done. Then we will generate the conformers by drawing the different conformers with the help of certain softwares. Then we calculate the descriptors. Then statistical methods or different methods are given. Through that we develop the equation. Then equation is validated and we predict the certain result from that. So what are the different descriptor we have? There are structural descriptor, electronic descriptor, quantum mechanical descriptor, thermodynamic descriptor. These are the certain descriptor which are available in the case of QSAR. So we will select on the basis of the result which we are assuming that we, if we want to assume or get the result, for that purpose we have to select accordingly the descriptor over via, through that we have to get the results. So you can see here one example I have taken, that here one equation has come out which is showing, which has come out, or which is the outcome of QSAR itself. That C is denoting here molecular concentration. Here these values are representing the regression coefficient. This is nothing but 95% confidence interval. Now you can see the parameter, this lipophilicity parameter, electronic parameter, constant form. So these are the different values. Like this we are getting the in the form of QSAR, and from this we will, when we calculate the different we design that model to the QSAR, and through this model we are able to predict the best proposed model, and through that we will develop the pharmacophore, and that particular pharmacophore is generally carried to the drugs and they to the lab, and in the lab its in vivo activity was established. So in the case of 3D QSAR, this is also advanced form of the 2D QSAR. Here we are going to have the equation, like in case of descriptor, we are having the van der Waals energy, electrostatic energy. Here the descriptor is somewhat different than that of 2D QSAR, and here we are having the 3D model set, because this 3D modeling set will not, this 2D shapes were having the limitation. Here that limitation got overcome with the help of 3D QSAR. So that is also an advanced form of the previous one. Nowadays 3D QSAR modeling is preferred over the 2D QSAR. Another is a pharmacophore based drug designing. This is one of the most common drug designing which the pupils are using, in which the, you can see the diagram itself by the blue color it is showing. We take the reference drug, over that the new drug which mimic the same structure is made to overlap over it. So from that we can design the drug. This is a like the dopamine structure is taken, over that one molecule is designed and try to overlap over that. So that will also give the hypothesis or the one of the way by which we can design the active drug. This pharmacophore generation process involves the certain step, that is the training set selection, feature selection, confirmation, then common feature alignment, then there's validation. So this is also one of the way by which we can also generate the pharmacophore. Then what are the different considerations we have to take, or the assumptions we have to take? That training set molecule which we are selecting here, it has to be diverse in nature. It should contain maximum structure information. That means, which already I have told you, that we must not confine to 10 to or 20 molecule. It should be have as much molecule as it can be, but it not be less than 40 to 50 molecules. Otherwise our study will be of the result we will get will not be fruitful. So we have to represent or take the molecule as much as we can take, or we have to select the training set in such a way that it has maximum number of molecules. So, and diversity is also responsible, because if we will not take the diverse molecule, our result will not be diverse, or we will not able to set out the result properly. So what are the pharmacophore features we say? We see generally hydrophobic character, hydrogen bond donor receptor activity, then hydrophilic activity, positive charge, possible ionizable negative charge. These are certain features we see in the case of pharmacophore designing. Like in the case of pharmacophore generation, we have certain input of the structures. Then through the input of the structure, we have the modeling, we have the conformation modeling with us. Then this modeling, through that model, we are able to have the model that model will be supported by the SAR data which we already having, and this generated model will give us certain hypothesis, and generated model through that we can evaluate it, or we can generate the pharmacophore. So this is one of the example of training set. You can see here diverse in pharmacophore is kept constant, and diverse structures are modified as per their requirement, and diversified training set, it can be designed like this. So one of the example from a paper I have taken, in that the training set is designed in such a way. So here you can see that this is the one of the example of the same which I have taken here, in which it is two rings we are showing by A and B. These are nothing but A and B can be substituted with the different heterocyclic moieties like pyridine or thiopine, and this substituent can have a variety of substituent, a variety of groups can be attached to that. And when these groups were designed or altered over this molecule, different structure we will get, and different structure when we get, through that we can have the uh, generate the pharmacophore over a reported compound. When we align it, maximum alignment there where we find, we can have that particular pharmacophore that we can take to the lab and can work over that. So this is one of the way by which we can also design the pharmacophore by pharmacophore selection model. This is a 3D model. Through that we are showing that it can be mapped. Like you can see the different hydrophobic moieties, hydrogen bond, and different van der Waals forces. Uh, these are the different parameters are seen by through this pharmacophore molecule is design. So these are the certain parameters we take into consideration while designing. Another way is receptor based drug designing. Receptor based drug designing I've already discussed. In this, if we know the receptor of the drug, so what we do, we just take out the receptor from the PDB ID that is already known to us. From there, we take out the crystalline structure of that particular receptor. Over that, when we just download it from the PDB ID, or from the protein, that particular receptor is there. So from there, when we just uh, align our drug or the design drug over the receptor, we can also have the drug, or that will also help us to design the new molecule. So this is one of the way of designing the drug, that is the receptor based drug designing, where we are knowing the biological target, and we are designing the drug on the basis of known biological target. Then another is the docking procedure. Docking procedure is nothing but the same way when we are having the receptor with us, and we are having certain area which is already known to us, that that is a particular specific area that is required for the activity. So we just take the drug, and we will try to bind the drug over that particular area, and we will try to minimize the energy by using the software itself, and we will try to keep the interaction as much as it can, and we will get the result in the form of scores. Scores are coming in the form of negative energy value, and higher the negative energy value, that means higher the docking value, docking score is there, that means more binding it is showing. More binding it is showing, that means the drug molecule is more effective. So this is also one of the way by which we can design the drug. So this diagram itself is showing that we are trying to dock the molecule over a receptor. So you can see that this drug designing can, how it can be done. So de novo drug designing say that it is based on the rational way how we can design it by starting with a smaller drug, and we can design by we are making the certain changes over the molecule, and we will try to see that how it is going to bind with the core or the active site. So you can see that from the starting, it has started with the 5-member ring, then it can, then this ring was added with this 5-member ring, and then it was tried to come into the pocket. So this seed structure is showing that nothing but the receptor, and the molecule which we are showing here, that is nothing but the selection of the drug molecule. So how we can design, how we can modify it so that it best fit in the receptor, That is the way by which we can design the drug. You can see here another is the receptor based drug designing and the pharmacophore based drug designing. So receptor based designing is known to us that we are knowing the receptor, we are just trying to align the molecule over the receptor. Pharmacophore based drug designing is based when we are knowing the pharmacophore over the pharmacophore, when we align the drug which we are trying to design. If they are aligned, then we can expect that they may be having certain biological activity. So you can see that these are certain way by which we can design. But when you will compare it, that pharmacophore or receptor based drug designing, so it require combination. We cannot align, or we cannot rely over a one thing, that is the pharmacophore or receptor based designing. If we will take both as a lead molecule, or both as a both we optimize both at a optimum level, then we get the ideal drug. So if, in the way we are saying that it may be ideal, or it may be the drug which may not fail further if we design it to the proper way, to the pharmacophore as well the receptor based designing. Because if we are knowing and pharmacophore is known to us, we can design the drug. If the drug is known to us, then we can design the receptor itself on the basis of the disease which we are targeting. So we can make the combination of both, and we can have the synergistic action by using the information we gather from both. So you can see here that in 1970 no biological targets were available. In 1990 we have the biological targets. Now we have the plenty of structured data of biological target, but also we have the improved technology to increase the pharmacophore based project. So you can see this nowadays we are having a lot of data with us. So this has reduced our time, as well as in another way it has also increased our responsibility to be vigilant that what is reported, what is not reported, on how it can be done, or how it has to be done optimally, so that the our drug molecule will not fail at the later stage. So when you will uh, see the lead discovery or lead optimization, that is not only the vague way that we have to design the lead and we assume that it may have the activity or not. We have to optimize it by using the different biological assay. We have to design it by using the different uh, techniques, molecular techniques which we have. We have to also use the pharmacokinetic modeling. Through that we have to, because most of the drugs fail during the pharmacokinetic studies, so that also we can design with the help of softwares. Pharmacokinetic profiling can be done, so it has to be also taken into consideration. So you can see when we have to develop the drug, and the drug is termed as a good drug when it is having the activity and the property optimization, then only it is termed as a good drug. Otherwise it may be termed as a good ligand, we have a good publication, but we will not maybe having a good drug that may exist in the market. So we have to also work over the property optimization as well as the activity optimization of any drug which we want to bring in the market. And drug discovery is not a based on a single parameter. It is a jungling. It is not a it's a dynamic process. If our drug molecule is active, it should be safe. Its pharmacokinetic profiling should be good. It should be novel. It should be soluble. It should be stable. It should be selective. If it is selective, then it should be stable. It should be active. So we can say all parameters are correlated with that, with each other. That has to be taken into consideration while designing any drug. And while profiling, we have to see these properties. They are responsible for any drug to come into the market. We have to see the different parameters which include the bioassay, metabolic profiling, whether solubility profiling, blood-brain barrier carrier, barrier solubility, permeability. These are certain parameters we have to also screen out while designing any drug. So we can conclude here that each stage of biology has a properties issue, that we have to identify the purity, solubility, then we have to see the stability, then we have to see the permeability, then we have to see when we are going to take it to the human beings, then also we have to see the metabolic stability, plasma stability, GI stability, solubility, permeability, PK, safety. These are certain parameters which we have to consider while designing any drugs. So, in silico admin models include nothing but different toxicity profiling as well as the pharmacokinetic profiling. So that will, if we work over that, it will reduce the iteration, it will reduce the rejection of the compound, which we are assuming that is due to the cause of failure nowadays. So, what are the different examples or the fruits of the QSAR? You can see certain examples with us. They are the results of the QSAR. These are the certain drugs which are discovered by the way the QSAR has come into the existence. So you can see here one of the few of the example of drug design successes. As we say, that drug design has a limitation, but apart from that, we must say that it has a rational, it has a utility, it has a certain fruitful results which are existing in the market. So these are certain drugs which are existing in the market, which are the results of drug design. In the last, we can conclude it's a drug discovery is a multidisciplinary. It is not a single task. It is a complex. It is a costly. It is involve the intellectuals. It involve the modern drug designing that make the drug more fruitful, and the a drug outcome or drug has come in a rational drug has come in the form of rational. It will come in the market rationally. It will come in the market. So knowledge management and technique specific expertise can save the time and the cost, which is the paramount need of the hour. So finally we conclude this that this is a way that we should know, or we everybody has to adopt while designing any drug, because nowadays it is desired. We must not go through the serendipity, we must not go through the older techniques, because they are costly, they are making they are costly as well as the time consuming. So we can reduce the time as well as the cost by using these tools or softwares of drug designing which we have. Thank you all of you.
Thank you, sir. Now I'm going to ask the participants if anyone can ask the questions, then you can write the question in your chat box. Sir, some of the participants wants to know which is the software basically used for the students, which is available in a free basis.
See, this is a common question the pupils are asking. Pyrex is a software which is freely available. Autodoc is a software which is freely available. You can have the tutorial itself over the YouTube itself. You just follow that tutorial and download it, and you can use it freely. There's a lot of softwares are available as a freewares.
Sir, also they also want to know the what are the learning apps for the docking.
See, I'm saying that if you want to learn the docking, you can have the YouTube videos. Through that you can read, you can learn the different steps that how you have to proceed. If you will follow it properly, because they are suggesting you from the initial itself that how you have to download, or what are the prerequisites to have the molecular docking software over your computer, what are the RAM you require, what are the basic requirement of your system, as well as they will also help you to download the software and how you have to implement it and how you can run it. So they have shown it with the help of certain examples itself, so you can easily learn through that itself.
So one question is from my side, sir. Sir, as far as we are using a natural polymers of the target molecule, but the natural target molecule having a many of the receptor site inside the body. So can we have any software or the pattern how to identify the target molecule can bind the particular receptor inside the body?
See, when I was discussing in the PPT, I have told you, then when the ligand is known to us, then it is termed as a uh, that is a ligand based drug designing. So that means you are not knowing the target. If you are not knowing the target, it is become very irrational that where you are going to bind the drug. So you have to first decide that the polymer which you are using, where you want to deliver your drug. So over that you can see whether it is going to bind over there or not. Because if you will talk about irrationally, that if we are taking any polymer and it has a multiple site over the body where it is going to bind, so our outcome become irrational. So you have to come with a conclusion in your research that where you want to deliver your drug. So you have to take that particular site. Through that site, you have to see what enzyme is there which is going to metabolize it. So you just take out that enzyme, that protein, and that optimize that protein. Then you just see the effect of the polymer which you are going to have over that protein, and you can conclude whether it is acting or not.
Any other questions by the participant side? Sir, one more question is that what are the different AI tools assisted to drug discovery process?
See, when we are talking about the AI is nothing but the artificial intelligence. So nowadays freewares are not available now, but that if you talk about the Schrodinger, if you talk about certain any we have certain Gromex, these are the certain software which are used, which are designed with the help of AI, because they are what they are doing, they are neglecting or they are minimizing your steps because of using their AI tools inbuilt in that. Otherwise, we have to go through the different steps. So that will be overcome with the help of that.
Is there any certificate course for docking?
Yes, a lot of uh, certificate courses are available over the docking. You just Google it, you will find a lot of courses are available. We also has designed one workshop in the previous year for the docking and the dynamics also, because now docking is just a preliminary stage. Later stage we have to go over the dynamic part. So that is desired nowadays. If you will see it, that only docking you will do, that will not be sufficient to publish your paper. Only that will give you the superficial idea that docking is done, then it may have the activity. Until unless the drug which you are going to use and which you are assuming that it is going to bind over the receptor, how much stable it is over the receptor, that is we get from the dynamics. So apart from this, we have to do the study the dynamics also. So you can see, you can Google it, that a lot of courses are available for the docking. So we have designed last year the one of the workshop for the 7 days. Now this year also we are trying to have this, so you can also join that workshop. That will be totally hands on. We will not going to have the lectures or the series of the boring lectures with you. Only the one or two lectures will be there, and the hands on will be there, so that you have the idea of that particular uh, docking and the dynamics.
So one question from audience side says that in Indian market, how many products are running that obtained from or that generated from the docking or the molecular software system?
See, I have given you some examples itself, that there are certain molecules which are the outcome of the drug designing. There I have given the example of donizepam, norfloxacin. So these are nothing but these are the certain example, or these are the resultant of the what drug designing itself. So we cannot say that it is not a it's a hypothetical thing. It has a utility. It has certain rational. Nowadays, people are designing through that. The COVID has proved itself that it has a rational. How it has evolved out. When you will see that this the drug designing is was there itself from the 1970s, but it has just evolved out in the COVID itself. Why? Because in the COVID itself has proved that we are not able to go to the lab. So from there itself, we have got the idea that it has a certain rationale or the utility, that we have to design the drug. We can design the drug to that, and the industries from the long back they are using it to design the drug.
So one last question. So estimated the binding docking software can estimate the binding score or affinity, showing the affinity on their software based. How it can be validated experimentally, or we can overcome the animal modeling, yeah, experimental process by the results of the docking.
See, the question itself has an answer. You are itself saying that when I was discussing or when was giving the lecture itself, then itself I have told you that whenever we are having certain results through the in silico, we cannot rely over that, because when it goes into our body, it has certain parameters or certain property that has to be also considered. So we have to optimize it through different softwares, because in that you will find there the GI motility is there, blood brain barrier is there, metabolic profiling is there, there are certain parameters are there. So if we have to justify it that whether our drug which we have designed is working properly or not, we have to justify it with the help of in vivo techniques. We have to perform the experiment in the lab in order to confirm the activity. Only the thing is that we have not to come with the thousand of the drugs. We can reduce it to the 10, 1, 2 or 10. Otherwise earlier it was there that we have to work over the thousands of the drugs which we are having.
Okay. So one question is another from the participant side. How do we address challenges the false positive and the false negative in AI based drug screening method?
See, when we talk about the false positive, false negative, you will see when any discovery is going to take place, it has both the thing. Sometimes we may having the result at the initial stage, but later we find that it is not working properly. So if you will see that that if we are totally based on AI, so we cannot say always the things are correct, because when you are using different software, different tools, when you will run it, you will get the result, you will not get the precise result, you will find that there are certain deviation the result itself. So we have to rely only when over that, when only we are getting the precise result. And if the result is not accurate, it should be precise, otherwise we will not able to rely over that. If it is precise, we can make it assured by performing different in vitro in vivo technique that result which we are getting is correct or not. Otherwise it is very difficult to depend on the AI itself to decide whether this drug is going to eliminate out this certain particular problem, because we know that drug designing itself cannot conclude that our designing of drug is sufficient to design the particular drug.
Thank you, sir. And I hope all the participants learn from this wonderful session. Now I am uh, inviting the Sarvesh Bhargav sir to give the vote of thanks for the our speaker. So over to Sarvesh sir. Now, sir, you can speak, sir.
Good afternoon to all. On behalf of Shriram College of Pharmacy, I feel honored to extend my heartfelt gratitude to Dr. Kamal Shah sir for delivering such an insightful and enriching session today. Your expertise and valuable thoughts have truly enlightened us and added immense value to this FDP. I would also like to thank our respected Principal Dr. Vinay Jain sir, Honorable Management and Organizing Committee for their constant support and encouragement in making this program successful. A special thanks to all the faculty members and participants for their active involvement and keen interest throughout the session. Once again, thank you, Dr. Kamal Shah sir, and thankful to everyone present here. Thank you. Thank you, sir.
Now it's time to present a attitude appreciation and acknowledgement certificate from the behalf of our principal sir, management, and the organizing team. So it's from the site from the entire team of our SRCP, Shriram College of Pharmacy. And we are very much thankful to you that you are providing your valuable time and your kind words to us and enlightening all the participants with the recent technology of the drug discovery and the designing, which is very much fruitful for the future generation of the molecules and the humanity and the welfare of the medical sciences. Thank you very much, sir, for being with us, and hope future we will connect with the same, sir.
Thank you, Ravindra. Thank you, all of you.
Thank you, sir. Thank you all the participants to join the meeting. I'm shared the feedback link in the chat box. Today's feedback link is present only in the chat box. We are not sharing the link in the group as well. ये बंद कर देना सर।